Hi everyone, I'm Bianca Lee from Cellular Agriculture Australia, and today I'm at the University of Melbourne Parkville campus where I'll be having a sneak peek into a cellular agriculture academic lab. First time the public has seen this space. It is one of a kind, built in 2016. And here I have with me as my tour guide, Dr Minh Ha, who's from the Faculty of Veterinary and Agricultural Sciences. Minh, what are we going to look through today?
Yeah, thanks Bianca. So first of all, welcome to Melbourne Uni, Parkville campus. We're standing in the beautiful system garden. So the tour I'll take you through today is looking at just snapshots of where we are doing research in cell ag. So cell ag is a production system, as you know, taking the meat from a muscle cell all the way to a final meat product that the consumers eat. And so we'll take a few snapshots along the way of which of those links we are actually working in terms of R&D on, and how do we take a single muscle cell and turn it into a beautiful juicy piece of meat that you might be eating.
Great, and so what are the different stages of that process that we'll be looking at? What are the lab facilities?
Right, so I'm guessing a few lab spaces that we're looking at today is the first one is where the fundamental muscle biology work happens, and that's where we learn a lot about the actual growing of the muscle cells. Fundamentally, what happens within the cells, what happens to the cells, what additives, what nutrients we're adding into the cells for them to grow. And then after that, I'll take you through another facility where I'll show you on the larger scale. So we're no longer looking at a cellular level, we're looking at a product level now. So this is where products are made, what happens to the products, how they package, how they are tested for sensory evaluation. And then I'll take you through to another area where we couple the sensory testing with the technical fundamental texture and flavor testing of the products to make sure that what we see in the rating of the consumers is actually explainable by the lab component of this.
Great, looking forward to it. Let's go.
Thank you. So we are in a wet lab at the moment. This is where we do a lot of our experimental work. Now, when we take cells from an animal, those are primary cells, we bring them back here and we start to nurture them. We start to add nutrients into them and grow from a few cells into a big mass of cells. And that's where we start the cultivation process. So the lab we're in at the moment is just one of the many shared facilities that we have in this building and indeed across the whole university. Now this lab here is shared by 3 different groups. There's the animal science group, there's the meat science group, and also there's a genomic group. Now what we do is we understand or we answer different parts of the production systems, and we bring different questions in and hopefully bring the same answers out at the end and improve that value of the meat product at the end by understanding different steps along the way. So come on through, I'll show you a few more spaces. Now by law and regulations, we have to keep certain chemicals in different spaces. Now this is an example of what a chemical room looks like. So this is where we keep all the chemicals to make the solutions, where we analyze the growth, the differentiation, the characteristics of our cells, and this is where all of those chemicals are made up, are stored.
Great, and so what kind of chemicals are we using to feed these cells?
So in the lab, there are a lot of non-food grade chemicals, so that can be quite dangerous chemicals that what we use to analyze the cells. But when it comes to the food production side, we actually use only food grade materials. So they are chemicals, but they are occurring naturally in the environment. So this is an example of freeze-dried muscle cells. What we're doing here is we're analyzing the collagen content in here and compare that to meat from a different treatment and see how they differ in connective tissue. And that would explain some of the differences in texture of meat. So some of the assays that we use in the lab to measure different characteristics of the meat, including intramuscular fat, so all the consumers understand that as marbling. Now, another type of assay we do is we measure the amount of connective tissue in the meat. Now, for a meat eater, they would understand that a top side is a lot tougher than a loin fillet, for example, and a lot of that is because of the connective tissue. Another type of assay we do measure is the sarcomere length. So different muscles, different muscle type will have different sarcomere length, and that will also influence the quality of a piece of meat. And so for Cellular, we need to understand how muscle cells grow into those characteristics, and because those will determine the quality of a piece of meat at the end.
Great. So this whole area is where all the magic happens. This is where you analyze how meat grows and how muscle cells and fat cells proliferate and differentiate.
Indeed, we do so using different equipment. So, for example, this is one of the heating blocks we use to incubate the muscles at different temperature for different assays. We have a shaker homogenizer. We have a microscope to look at the muscle cells under different growing conditions and see how they behave differently, how they respond to different nutrients differently. So now I'm going to introduce you to one of my honor students, Brodie Peace, who is working on the media component of how to grow these cells using different food grade ingredients and stimulate the cells to grow.
Hi everyone, I'm Brodie Peace. I just finished up my honours degree here at the University of Melbourne, and what I'm going to show you today is how we culture muscle cells to make meat. So here I have a cell sample of C2, C12 cells. Now that means they're a mouse skeletal muscle cell. And what I'm going to show you is my lab, and we're going to get these growing. So the first thing that we need to do is take them out of this cryo vial, which was stored at 200 degrees below zero, and put them into some growth media. So I'm spraying it with ethanol to kill any contaminants that might be on the surface of the tube. And now it's in this biosafety cabinet, which has a fan here that is extracting anything from the air and sucking it underneath to prevent any contaminants that might be in the air or in my breath or even falling off my skin from entering in the chamber and contaminating the cells. And here I have a flask that I'm going to put the cells into so that they can start growing and multiplying. So now we have the cells in the flask, we need to add some growth media. Now, cells, unlike animals, can't just eat food, chew it with their mouths and digest it with their stomachs. They need their food in the rawest form, with all the nutrients in liquid, so that they can absorb all the nutrients to grow. So inside the growth media, we have amino acids for the proteins. We have salts and sugar, as well as animal serum, which contains the growth factors that signals to the cell they need to grow. So now I've added media to the cells and we're going to spin them in the centrifuge. Now this is to separate the toxic freezing media that's needed to freeze the cells from the cells now that they've been thawed. Now that I've added media to the cells and I've swirled them in a figure-8 shape, I'm going to leave them there so that they can attach to the surface of the flask. Because like in our body, muscle cells like to grow on the bone. They grow attached to something, and so we're allowing them to attach to the plate. Now it's been about 15 minutes. So the cells are ready and attached to the surface. Let's have a look. Yep, growing nicely and ready to go into the incubator. Now cells don't grow in the normal air that we breathe. Cells need to grow like they would in the conditions as if they were inside the body, which means at 37 degrees Celsius and 5% carbon dioxide. So this is an incubator which has a gas of CO2 leading in to make sure that it's at the right level, and it's heated to make sure that the cells are at the right temperature. In 2 days' time, when the cells have grown, they'll be ready for a media change to get fresh food so they can keep growing. So once the cells have grown and we have enough of them, and it's time to analyze their growth and the different elements that we want to examine, we can then use stains to stain the different parts of the cells. So if we want to have a look at the DNA, we can use this hematoxylin stain. Or if we want to have a look at the muscle fibers and the structural parts of the cell, we can use this lighter stain called eosin. So, in the image, you can see small purple dots. That's the nucleus, which contains the DNA of the cell. Those were stained with hematoxylin. And the surrounding parts, the myotubes, the longer structures that you can see, are the muscle tubes which have been stained by the eosin.
So now we're going to meet one of our PhD students who's working on cultivated seafood.
Hi everyone, my name's Kieren and I'm from the University of Melbourne. I'm doing a PhD on the characteristics of seafood so that companies creating alternative protein products can mimic them better, like the colour and the texture and the taste and aroma. I'm going to be showing you the texture analyzer today, which companies can use to check that their alternative products are the same texture as what they are mimicking. And we've got to make sure that the texture analyzer actually measures like a human would measure with their finger or their mouth. All right, let's get started. So this is a Lloyd texture analyzer. And so it's got a probe down here, which cuts or shears the sample connected to the load cell, and the load cell can go up and down. And so as it goes down, it measures the force it takes to go at every centimeter through the sample, or actually more precisely than that. So let's give this a go with raw salmon. We're going to start the measurement now. So the probe is going to come down and the machine is going to measure how much force it takes at every millimeter to go through this sample. All right, let's have a look at the results. So this is the Warner-Bratzler hardness. So as the probe came into the sample, it required more and more force to be able to cut into the sample until finally fracture, and that is the Warner-Bratzler hardness. And that should correlate with how it feels in our mouth and how much energy it takes to actually break the sample. Data on the texture like this is really important for companies creating cultivated meat. For example, they could use this machine in the same way to test their cultivated salmon to check it's the same as animal-based salmon.
All right, we've got our hairnets on, our lab coats on. What is this room and what is all this equipment?
So as you can feel, it's very cold in this room. It's always set at 6 degrees. Now this space is where we do a lot of product formulation. Now with products, so as you remember upstairs where we took a lot of our cells, now a lot of our different products, we bring them down here, and this is where we formulate what the consumers will see when it hits the supermarket shelf, right? Now, one of the things is, so this is the space where we do a lot of formulation. This is where patties are made. This is where sausages are made. This is where all different blending of perhaps even meat and plant proteins together. Now, once a product is made, they then have to be packaged. As you know, when they hit the supermarket shelf, that's where different packaging systems will affect the eating quality of that product. So this is an example of a vacuum packaging. This is where you see for large cuts of meat, this is what happens. Now, if we are growing a full muscle cell, for example, in the lab, they will need to be packaged in here for transport across states, across countries, and also in retail as well. If I take you through here, one of the most common form of packaging that you see in supermarket for meat is what we call modified atmosphere packaging. Now, these are where meat sits in trays about yay big, right? And if you see the plastic sort of curving up a little bit again in that tray,
Yep.
that is modified atmosphere packaging. And this is the machine that does that. So, and our research at Melbourne Uni have shown that different packaging systems affect the biochemistry inside that meat and then affects the eating quality of that piece of meat. So for cellular application, of course, the meat products will eventually have to be packaged for retail, right? And so understanding how the different packaging system affects the meat cells and affects the quality of that cell is important for different companies and the consumers. So once a product is made in this space here, then it has to be shaped into whatever final form that we're looking for, whether that's a sausage or a patty, and cooked for analytical methods of texture measurement, for example. So this is the process meat lab in here. This is where a lot of that final product formulations are made.
Let's go.
So this is an example of what I mean by product formulation in final shaping. So this is a bow chopper. Now this is where we put the meat in and chop it up into small pieces. Right, now that could be from animal or could be from a lab-grown material, right? They go in here and then different ingredients are added in there. Well, those ingredients will decide what shape and what type of products the final products will have. So this is a sausage stuffer, for example, right? This is where we'll take the mixture from the bow chopper here, put that in here, and the shape of that final product is then coming out from here. And that's where we take the products and we put them into a cooking equipment, such as this water bath here. Now, this is not just a water bath where you can just plonk it in your kitchen.
It's not a sous vide machine?
No, well, actually, no, it is a sous vide machine, but a scientific sous vide machine. So it costs quite a bit because the precision in temperature control is extremely high. So we're talking plus minus 0.1 degrees with this equipment here, yeah? So with this equipment, we can do all different types of cooking and see how different types of cooking, how different temperature, the end temperature, as well as the increase in temperature over time, how that affects the muscle cells, how that affects the fat cells and the connective tissues, and how different cells start to denature or relax is another term that consumers say it. How does the meat relax? And that's what happens in here. So this is where we really understand that chemical and fundamental side of it.
Great. So you said different types of cooking. What are some examples?
So in here we can do open air cooking. So it's where the bags are not sealed, or we can do sous vide cooking. So sous vide, as you know, is a French word for under vacuum, right? So
that's where,
right, there you go. So that's where we vacuum pack the piece of meat, or in this case, a piece of salmon, and then we put it under circulating temperature at a certain temperature with, as I said, plus minus 0.1 degrees. And then we analyze the texture, the flavor, both with the consumers as well as with equipment, and understand what kind of texture, what kind of flavor that piece of meat or that piece of salmon in this case has.
Great. I want this in my kitchen.
Now, if you remember, Kieren Watkins was analyzing the texture of salmon. Now, what Kieren's doing is trying to objectively measure that texture. Now, how do we really know what the texture is really like? By asking the consumers to rate it. But of course, before we ask the consumer to rate any kind of meat or seafood product, we have to cook it first, right? And so now we've got the food sensory kitchen. This is where we do the cooking, not for testing with machines, but for testing with consumers. So,
My favorite part.
that's right. Come on through. So now, temperature is really important to meat products as well as seafood. Now, Kieren was testing texture of seafood cooked at a particular temperature with the texture analyzer. Now, if you transfer that over to a piece of a commercial or a domestic kitchen equipment and cook at the same temperature, but now without the scientific setting, how does that piece of salmon taste? This is where we really find out here. So this is a piece of commercial oven, but with high scientific setting, where we can really test the temperature at different endpoints, at different profiles, where we can start low and ramp it up high, or the other way, start high and ramp it down to low. And that's really trying to understand how the proteins in meat behave and how the consumer then respond to that behavior. Now, another piece of equipment, and this is no normal barbecue at home kind of equipment. Right, now here we've got what we call Silex grill. This is again a scientific equipment for meat testing. So this is again where we are grilling meat, but at a very specific temperature setting. And there are two different plates where you can set the plates at very different points to understand how the temperature difference in different parts of a piece of meat might influence the consumer liking of that piece of meat.
Right, so this is not your average toasty in the staff kitchen.
No, I can tell you one of these is worth about a tenth of your normal barbecue systems at home. Now, once we cooked a product, we bring it over here for the consumers to tell us what they really think about it. So come on in. So this is the inside of our consumer testing booths. So this is where we are serving a product to consumers who are sitting on the other side of this booth. So by having two different sets of doors, we are presenting to the consumers different kind of products at different times. Now there are 20 different booths in here. So this is where a product is pushed through, and there's a button here where we can let the consumer know that the product is ready for them to consume. So now let's have a look to see what the other side looks like, where the consumers are sitting and testing a product. Now in the old days where we still use paper-based methods, these days we use these touchscreen methods. Now this is not just a normal iPad, it's also got the capability to measure a consumer's temperature, heart rate, physiological response, and even the movement of their eyes, and see which part of the product that they are really focusing on, because that's important for us to understand what is it about the product that's really attractive to the consumer. So we are now looking at a GCMS, that stands for gas chromatography gas mass spectrometry. So this is where, if you remember, I mentioned flavor before, right? We test flavor with the consumers, but this is also where we actually extract each volatile out and ask a consumer to sniff the gas that comes out and say, tell us whether that gas is buttery, or is that umami, or is that floury? And so to understand that complex mixture of gases, of aroma from meat, this is the machine that does it.
All right, Minh, so you've taken us through the entire process of how to grow muscle cells to create meat in the wet lab. Then you took us to the meat processing side, where we see how meat is broken up, how we can measure quantitatively the texture of meat and how we can package it. And then you took us to the consumer end, where we can actually measure how consumers perceive and taste and enjoy meat. It's been really fun. There's no facility like this in Australia, and your lab is one of the handful of labs in our country that is working on cultured meat research. So thank you again for today. It's been awesome, and I'm really looking forward to seeing what your lab does in the future.
Thank you. And look, we're always open for engagement, for interaction with the public, with potential collaborators, people who are interested to know feel free to email me. I'm on the website. I'm on Farm Expert at Melbourne Uni. Feel free to get in touch with me. I'm more than happy to talk.
Great, and of course your details are also on the Cellular Agriculture Australia website, where we've got a list of all the labs and all the companies in Australia working to produce cultured meat.
Thank you.
Thanks, Min.
Bianca.